Then we will proceed to the next speaker, Jorg Rau. He's from a state chemical and veterinary investigation office in Stuttgart, Germany. And he will give his talk on spectrometry-based technologies in public health.
Okay. Excuse me. Thank you for invitation to give a talk about spectrometry-based technologies in public health with a focus on microbiology and microbes. Okay. As introduction, I want to give an overview of the governmental labs in Germany dealing with differentiation of microbes. And I will show the rising role of spectrometry in our world in the meaning of vibrational spectroscopy and the mass spectroscopy methods. Some aspects about workflow, validation, and a nice example from we have recently made a little prevalence study and where we can combine both methods. And at last, I want to close with some expectations in the future. What's the scope of governmental control? We're dealing with all aspects of human health, with animal disease, especially with focus on zoonotic microbes, and with all things of food and water control. So in Germany, only in the food control, 400,000 official samples of foodstuffs are investigated every year. This task, this food control, is the task of the federal states of Germany, not from the nation at all. So, in most of the states in Germany, more than one lab was dealing with some aspects of this field, and so we get a real landscape of laboratories with different specializations. National reference labs supplemented this system. For animal disease, food, here in Berlin, the Federal Institute for Risk Assessment, or the Robert Koch Institute for the human health aspects. Now I want to focus on my state, Baden-Württemberg in the south, where we have 4 special labs for chemical and veterinary investigation offices, 1 state veterinary laboratory, and a special lab for the public health aspects. Here is Stuttgart. It's my site where I came from. What were our samples normally? We are dealing with everything with hygiene monitoring, water and foodborne food control microbes. We have to investigate foodborne diseases from the patients to the food, what is eaten, the ready-to-eat foods, back to the equipment, maybe the kitchen staff or work environment thing or raw food. The animal health side is dealing with all things with animal health monitoring, health service, and pathology samples around animals. And human pathogens are concentrated in this State Department of Public Health. In the next chapter of my talk, I summarize some of these basic methods we use in the detection of bacteria in our labs. It's in the most labs the same. We have the first step, a direct culture. We use selective media for some food pathogens to obtain the isolates, and here was the starting point of any identification. Classical ways for this are phenotypic characteristic biochemical profiles. They are still alive in these labs. Genetic characteristics, special PCR techniques, which analyzes genetic features of some special target microbes. And these methods still build the status of the official methods we have in food and water microbiology. As an alternative, spectroscopical methods are introduced in this world. We use, at first, it's the site where I came from infrared spectroscopy. We use in our lab since near 10 years. This technique was reintroduced by the Robert Kopp Institute nearly 20 years ago in our field. And in the next step, we recently introduced Malditov mass spectroscopy 2 in this biotyper mass spectrometry methods. Okay, infrared spectroscopy uses the infrared light. This is absorbed from different biomolecules in a special way and in different ways. So you have specific markers for some specific chemical components, like lipids, amides, and polysaccharides. And sometimes you have a real fingerprint that you can identify isolates. This infrared spectra you can scan for characteristic patterns, for genus, species, for subspecies, and sometimes for some factors of pathogenicity. These patterns are like an image of the total chemical composition of the sample. In a very easy example were the differences in the composition of the cell wall, which are reflecting in this lipid region, and here are the specific markers for this differentiation. Malditov, on the other side, was used clear mass differences, mainly from ribosomal proteins, so that you can reach a very precise taxonomic marker system. That it's a differentiation in genus and species level was able. In our lab, both methods have a very similar workflow. Starting from the pure culture, in the infrared way, a preparation on a sample target was done in a zinc selenide plate. A drying step was obtained, and these bacteria films were introduced in the infrared spectrometer. Here it's a Bruker Tensor 27 machine, and then this spectra were identified with a special database. Mass spectrometry is like the same. You have pure colonies, not cultures. You need much lower amount of cells. The preparation is done on this sample target, the matrix is added, and you get a result in a very short time. Infrared spectroscopy needs much more time in preparation, 2 hours maybe, and you need standardized culture. In our case, normally we use 24 hours. In the case of Malditoff, sometimes you can dealing with 8-hour cultures because you need only a little amount of cells. Before, the workflow in our lab was dealing with the samples from different sources, food, water, animal, or some external samples. They were tested in this standard procedures for our microbiology, and then these isolates in the next step were verified by infrared spectroscopy on species level, but also on subspecies level and sometimes for investigation of contamination routes. We used here our own database, our own isolate collection to feed this database, and this system was supported by our PCR group who deliver us data for all the sequencing data, yes. All this data are from the specific tests are going in our LIMS system at all for the opinion and so on. Now the workflow is changing very rapidly when we introduce the Malditoff. The first step is now the Malditoff. We combine it. Malditoff, most of the bacteria are differentiated with the Maldi, and in the second step we use FTIR for subspecies or for this contamination routes. And some species are, especially in the food context of Listeria and Bacillus cereus group, we still leave in this in the infrared because we have up to now better experience with it. And in the second step, we will introduce a little own database to combine new strains from our isolate collection, especially from animal microbes. Now we have a new center for our differentiation, and but it was a new method. In governmental labs, you have every time the same question when you want to use new methods. Spectrometry is not accepted of the official method in a special German food and feed law. So we have do a special in-house validation to prove the methods. So to select this special for validation, we analyze our strain collection and look to species of interest. And so you can see that 50% of all of our isolates we have stored are come from dealing with food and were from food-borne pathogens, Clostridia, Yersinia, Salmonella, and so on. So we do a little validation process for our important parameters, which should mirror our world of bacteria. So strains from the isolate collection are differentiated by the Maldi, and a standardized validation protocol was used. We have used before for the FTIR section with this little validation table, and we have to use a control group which is nearby the target bacteria. In this example, when we have the Staphylococcus aureus, we need other staphylococci for the control group. In this questionable results, species okay. So we see that we have now an extended toolbox for our microbiology. The classical methods, PCR, some serological methods, and MALDI-TOF and infrared spectroscopy. So we can do many things we cannot do before in our routine lab. I want to give a special example. We have a collaboration study with the VetNet lab in Ludwigsburg. It was an animal health lab. In this study, we have investigated Yersinia enterocolytica in fecal samples from dogs and cats. After enrichment and isolation, it's all done by the IDEX. They screen and primarily identificate the Yersinia by MALDI-TOF. So they get 187 isolates. So we get a prevalence of 4.6 for dogs and 0.3 for cats. And all these isolates are sent to our lab that we make a second investigation with infrared spectroscopy. Here we make a confirmation and a characterization by the FTIR for biotyping, and the IL factor, it's a special factor for pathogenicity in Yersinia enterocolytica. All this data were verified with classical methods and with the PCR for bioserotyping and the IL gene. And then in this last step, we compare the infrared spectra in such a cluster analysis. And in this comparison, you see very clearly that the biotypes were found in special branches. And the IL-negative biotype 1A was clearly separated from all the other ones. So we see a powerful combination of MALDI-TOF for reliable identification and FTIR for further subspecies characterization, resulting in a fast workflow. And for our microbiology, it's a very new and opportunity. Now, in Baden-Württemberg, the federal state, we introduced, or we bought 4 MALDI-TOF machines recently, and then two sites we have infrared machines. We want to get in intensive cooperation. We get more interdisciplinary work, assistance between the labs in Baden-Württemberg, and an internal backup system if one machine is gone or so on. We want to share the validation data and the quality things. We have our own database section, and we made some external cooperations. We expect a broadened application for food, animal, water, and so on. The reliability of the methods will be more and more growing for bacteria, yeast, fungi. We see it in our own validations. We get a much more faster analysis of all this, and we get a better acceptance for these techniques because it's open for extension. We can do our own database sections, other target proteins, and we get a powerful combination with our FTIR technique. Okay, and then I can thank you for your attention.
Thank you, Dr. Rau, for your presentation. This lecture is now open for discussion. Are there any questions from the audience? Please go to the microphone. And maybe I will start with a question. There are several papers on typing pathogens with MALDI-TOF for outbreak investigation. And you showed us that, that this infrared spectrometry is also used by you for outbreak investigation. Did you ever try to compare these two systems for this purpose?
No.
No.
So the next project to go, probably. Doctor, one question I always have is about FTIR, this infrared spectroscopy, looking at the whole cell content, needs more standardization, everybody knows, but what do you think about the possibility and the limitations of sharing libraries in this area? Has every lab to create an own library, at least for subtyping, or is this something maybe at least in Baden-Württemberg you can share?
The FTIR are not so, it's only a very exceptional method in this field. In Sigmaringen, the second one is a second machine. So I have now many experience with transport database to another site. And so I think, so the MALDI-TOF, it was shown that the commercial database will function in every site. So I think it's...
So we wait until we get results from you before we decide if we do it otherwise. Huh? Okay, thank you.